Hello, everyone. My name is Laura Furlong from Biomedical Informatics, and today we have Alba Gutierrez Sacristan from Harvard Medical School. Hello, Alba. How are you?
Good. How are you? Good morning. Thank you for having me today.
Yes, it's a pleasure to having you. So, would you like to tell us a little bit about yourself, what you do at Harvard Medical School, what is your role?
So, my name is Alba Gutierrez. I'm an instructor in biomedical informatics at Harvard Medical School in the Department of Biomedical Informatics. My background is in bioinformatics and biomedicine, and my role here right now as instructor, even if I started as a postdoc, right now includes research and teaching.
Great. And as we are going to talk about DISGENET today, I wanted to ask you first, how did you know DISGENET in the first place?
Yeah, so I think I grow as a researcher with DISGENET because it was during my PhD when I came across first with the platform. DISGENET was being developed by the lab where I was doing the PhD, and it was great to see how this was being built with multiple research and experts in different fields, including biomedicine and natural language processing, and being a little bit part of it. And yeah, since then, I think DISGENET has been always part of my research, a tool that I always come back to search, and also a tool I use for my teaching here.
Great. And how do you use it for teaching or in which subjects? Can you tell us a little bit about this?
Yeah, I teach here several courses in use of databases, but also in system biology, omics data analysis, and integration of clinical and genomic data. So it's part of the learning process to see from where the data is coming, where are the pre-knowledge on a specific gene disease association, for example, to do a PheWAS. So we introduce many different databases, ClinVar, UniProt, PubMed, and we go with the students through the logic of the steps that you would follow in when doing a research. And I think one of the roadblocks that we find is when you need to do searches in multiple databases. That's why DISGENETs come handy, because it's like you see many of these issues that we are seeing when trying to integrate data of different public databases is already solved here. Plus, I always put this example to them on how long it will take you to read all these papers that you have in your to read folder that you will never touch back. So we also see how helpful it is to know NLP and all this, but also to use databases that already have done the work for us. Like, I think being efficient is also one of the key that we tackle on my courses.
Sure. And can you tell us a little bit more about specific projects or tasks that they have to do and in which DISGENET is found useful or they can apply DISGENET?
Yeah, so for example, one of the courses we perform differential express gene analysis. So after all the analyses are done and we have identified the findings, we ask them to identify if those findings are novel or are previously described. And one of the steps is to go and check in DISGENET for the knowledge about it, but also check the first time that this has been described, the last time, how many times. So this can give you a lot of points and hints on it's novel or no, it's super reported, it has been studied a lot. That would be one example. The other one would be when doing phenome-wide association analysis. This is exploratory analysis to identify potential associations that have not been previously reported. And the original description of a PheWAS is starting with SNP. So, for example, in one of the other courses that I teach, what we ask them is to investigate the specific SNP and see all the phenotypes that have been previously reported associated. And then they run the PheWAS, and then they always have to go back to DISGENET, for example, and other databases to see, okay, the findings they have were already reported or not, to identify potential novel findings for them to continue doing the research after the PheWAS is done.
Interesting. So they are using both for finding or analyzing genes associated with diseases and also to identify variants to then perform a PheWAS analysis.
Yes,
And
both ways.
Okay, great. Good to know. And for identifying, for instance, variants, or do they found something useful that DISGENET offer to, I don't know, prioritize or better select the variants that then they will use for the PheWAS analysis?
Yeah, they found it super helpful because it allows them to just center the focus and the search in one database, and from there they can move. For example, they in the specific case of the PheWAS, they were also interested on before actually extracting information from the BCF files and analyze the genomic data to see what was the expected allele frequency for that SNP. So having the allele frequency helped. In their case, they had to also complement it with the genome browser because we were working with TopMed data, but having an idea of how in Nomad this was reported, this definitely helped them to prioritize and say, okay, yeah, this is an interesting SNP, but the allele frequency is so low that we will not find potentially many participants on the study we are analyzing with it, and then the statistical power is not going to be enough. So that helped them refine that a lot, and also to have the overall idea of where it's located, what information they are going to need, and get a sense of, based on the sentence of the manuscripts, get a sense of what has been said about this variant and this specific condition. So was super helpful for them, and they found it again like an efficient way. Like we are always trying to save some time of exploring multiple things, and here it was like all together.
Great. Good to know. And as you know, DISGENET offers several tools to work with the data, web interface, REST API, R package, Cytoscape app. Did they use in particular one of the tools or all, or how do they interact with DISGENET?
Yeah, the first interaction is always with the user interface. Go to the browser, do some search. This really helps them to better understand how the data is collected, especially when it's the first time they are interacting with the database. Once that has been done, there are I have two profiles. Some students that feel much more comfortable just using the user interface, and they will continue just with that. And we have others that feel more like tech, and they feel more comfortable with the programming part, and usually they go and use the R package, so they integrate the functions and add some visualizations to their pipelines in an easy way, because all our coding in our courses is done in R, so it's an easy way for them to integrate all this information.
Do they find the documentation useful enough, or do they miss something?
They usually ask a lot of questions when something is not clear. And in this case, I think it was very self-explanatory, like especially the user interface, because you can just put your mouse on top of the things and get some more detailed information. Probably, I think the part I got more questions always was about the scores, because they are not that familiarized with that and can be a bit confusing maybe the first time. And it's great documented, but I think it always helps for them when you explain them a bit more step by step. And they found really interesting the way that was done, not only taking into account like how they can rely on the score to say, okay, this is something reported in many places and many publications versus others, that not just based on one number on the scores.
Good. And if you had to like suggest improvements or something that you would like to see in DISGENET that is not currently available, or something that could be improved?
Yeah, I think the R package, I have been following it, so I have seen the new implementation and the new functionalities, and some of my wish list, to say in a way, I see that have already been developed and implemented. I think the part that I still find challenging and I cannot find also in any other database is more on the disease association, that we work also a lot, that it would be great to be also able to get some information from papers and get more additional information in the same way that is for the gene disease and the variant chemical and all this, to have it for the disease, and also additional information on the more on the NLP part, to say in a way, because it's true that once you have the information of the papers, one of the main questions that many of my students have been asking, and also myself when doing my research, is like, what is the population in which these analyses have been been done, because that also can help you to classify the articles. So the answer for me is always the same. They need to go and read the papers or by themselves apply some NLP, but it could be, if you ask me for a wish list, that would be something that would be awesome to have in how many participants, the characteristics of the participants, the country, the sex ratios, like some additional information on the population, because the population characteristics really have an effect on the gene disassociation. So I think that's something that I would love also to see there.
Okay, we take note for future releases. I mean, we are working in some aspect of this, collecting ancestry, especially from GWAS analysis, and that the other things that you mentioned will be more challenging, but it's worth to try to capture this data. So, and you also mentioned that you applied DISGENET in your own research. Can you tell us a little bit more on how, well, first, what you do in your research and in which aspects do you apply DISGENET?
Yeah, I do a lot of comorbidity studies. I work with electronic health record data and claims data. So even if now on the electronic health record data is more common and frequent to find genomic information, it's not always the case. It's not the standard. So sometimes when doing some comorbidity analysis, when we want to explore further, okay, why we are finding these two diseases together, is for me like a step that I always go back to DISGENET and say, okay, is this reported? They have something in common from the genetic point of view. So that's one aspect in when I use it a lot. And on the other hand, I also perform many phenome-wide association analysis, not always with genomic data, but sometimes I do. So when including some variant information, again, I always go back to DISGENET to see, okay, with which variant I could start that could be a good example, or maybe even just to develop my code in a way that makes sense. So I just check something that is not just random, that is based on something, even if it's commonly described. For some steps, I'm not that worried about that part. And also in our lab, it's all about integrating clinical and genomic data. So we have information from many databases. So when we need to develop also case studies that make clinical sense, that's also a resource I use a lot. All day, it's on my tab, common tabs that you go back to.
And if you had to recommend DISGENET to other professors, or teachers for different courses, what would you tell them about DISGENET?
I think my main advice would be just try, because I think as a researchers and as a teachers, we are very used to just go through the things that we feel comfortable and we know about, but we should never stop learning. And when you learn a new tool, then you realize that maybe you could be saving a lot of time, even if you already have a great pipeline developed before for the same. So I would just say, just try it, create an account, do some research. And I would say also check one of the 1,000 publications that reference the use of DISGENET to see the potential that it has, because it's been applied a lot on research. So I think sometimes when seeing what others are doing can also give us some ideas of, oh, maybe I was doing this in this other way. So I would say just go and try. Every time I have an opportunity, I always say, have you tried DISGENET? Especially because, again, like the information is really dispersed in many different databases. So when they told me, oh, I have been doing a search about this variant and here and there and there, it's like, did you check DISGENET? You could have get this done just in one search. That's the way I would emphasize this. We all like to save time. We all like to be efficient. So go ahead and try.